Hi, good afternoon, good evening, everybody. I'm Moshe Weinstein. I'm CEO of Everads Therapy. I'm here to present our suprachoroidal delivery technology, and I'm excited to be part of the OIS and to present some of the great technologies coming out of Israel. So who are we? So first, as many of you are here also, we are here to restore vision by enabling optimal treatment of debilitating retinal diseases. Our focus is around suprachoroidal delivery and the belief that the suprachoroidal space is an advantageous delivery route for treatment of retinal diseases. And our differentiation, and we'll go into this during our presentation, is really the safety, the rapid distribution, and the ability of our technology to deliver high volumes and large particles that will rapidly distribute throughout the posterior pole and the macula. So what is the suprachoroidal space? I'm sure many of you are familiar, but for those of you who aren't, we're talking about the space between the sclera and the choroid, including the extravascular space of the choroid itself. The space circumventually spans the entire posterior segment of the eye, and it's often referred to in literature as a potential space or a virtual space because it gets opened up or created as fluid is injected into this space. It's an emerging route of administration for ocular disease treatment over the past decade, and there's an increasing number of preclinical and clinical investigations going on as we speak, where we're using the suprachoroidal space as delivery. And those are in gene therapy, for steroids, TKIs, and even anti-cancer agents. So why all the interest in the suprachoroidal space? So delivering to this space allows you to directly target the choroid and the retinal tissues with posterior and a circumferential spread. It's really an effective way to reach the posterior pole and the macula. And when you inject through the space, it doesn't interfere with the visual axis, really targeted. It minimizes risks of off-target effects. There's a lot of speak of using this space as a potential reservoir for the slow release of therapy. And again, it's really ideal when the efficacy or safety profile is not acceptable when treatment is given intravitreally or subretinally. There's, of course, a freehand approach, but it is very complicated and often unpredictable and not really repeatable. And there are just a handful of technologies that exist for optimal delivery to this space. What is our technology and how is it differentiated? So the Everads technology is really around entering the eye tangentially and using a tissue separator that's been designed specifically to open up a channel or a space between the sclera and the choroid and allow you to inject into the suprachoroidal space. And I'll show you an example of how this works in a video coming up. The injector itself, as you can see here, from the tip of the needle basically extends this tissue separator and retracts it, done in a matter of a second. And through there, we've opened up this virtual space, or just the first mile of this virtual space, in order to inject. This is just sort of how our kit is delivered. It comes with an injector and, of course, a Lure-Lock syringe. We can work with any standard Lure-Lock syringe. And it has a fixation tool that's used to hold the eye in place as we're giving the injection. So the best way to really understand this technology and the distribution we see when injecting suprachoroidally is to see a video of the procedure itself. So this is a procedure which we're doing using ICG in a New Zealand white rabbit. We use, as you can see here, the scleral fixation tool to hold the eye in place as we advance with our device, inserting the tip of the needle a few millimeters past the limbus. We insert the device to a controlled depth. There's a stopper there that's optimized for the right depth to enter. We flatten the needle, and what you don't see here, you could slightly see it under the sclera, is the extension and the retraction of the separator. That's done, and what's happening behind the scenes, because you can't see, is that the operator is injecting ICG, pushing down slowly, injecting your 150 microliters of ICG, and as it's injected, slowly releasing the fixator tool, and then he's going to hold the device in place, the needle in place for a few seconds, pull out, and you'll be able to see the reflex of the green. So here, the operator pulls out, you can see very little reflux coming out of the injection spot. And you can see as the eye is moved that the reflex of the green illustrates, you can see that we're in the suprachoroidal space. You'll be able to see here the distribution. This is covering probably 80–90% of the posterior pole. You can see here there's ICG of both sides of the optic nerve. And we really get a great and almost immediate distribution. So these are videos that are done minute or two post-injection. We have experience also doing this in non-human primates. And you can see here OCT images, which show highlighting of the ICG dye, and they show the wide distribution throughout the macula and optic nerve. On the left-hand side, you can see one eye where the lit up spots are actually the parts where ICG has reached. So you could see in this eye, within 1 hour, 3 hours, you're really up to the optic nerve. And at 24 hours, you've covered almost the entire optic nerve in the macula. And then in the other example, actually, you don't see a dark space. In this specific example, we've injected 200 microliters. You see a complete distribution almost immediately of the macula and the optic nerve. So how is this technology differentiated? So first, we've really proven the ability to quickly reach the back of the eye and the macula, right? We're able to deliver large volumes, 150, 200 microliters, and different from other technologies, there's no bleb that's formed upon injection. Really, we see with this tangential insertion and injection, the test articles are distributed very quickly to the back of the eye. With this technology, we're able to deliver large particles and even viscous materials. And because of the way we're entering the eye, we have improved safety because we have very low susceptibility to variations in scleral thickness, right? We have the flexibility to inject closer to the limbus, right, and remain quite interior because we're using this novel tissue separator to open up that last mile and extend past. So there's no need to enter the back of the eye with sharp tools or feed a cannula. As you saw in the video, there's limited reflux as the result of the way we enter. And of course, we're able to adapt our technology to work with standard Lorlock syringes. We have experience with different modalities. We have experience with viral vectors, with protein-based therapy. We've also delivered gels and viscous materials, and our device has acceptable injection forces to deliver these. Our inventors have done a lot of work on stem cells and microparticles. And we have experience in non-human primates, in the cyno monkey, in the African green monkey, in New Zealand rabbits, and in pig, all of which we've been able to show a safe and effective distribution. What is our strategy and how is Everads bringing this technology to market? So we are seeking co-development opportunities with pharma and biotech companies who have therapies that are ideal for suprachoroidal delivery. These could include gene therapy or cell therapies, novel anti-inflammatory agents, or other sustained release therapies. We've already signed an option deal with a large pharma company, and that deal is going quite well. I can tell you after 3 months, we received payment and they exercised certain rights to have exclusivity on specific retinal targets. Of course, there are a lot of retinal targets out there, and we're really looking forward to forming additional collaborations with other companies. I must say it was an honor to be a part of this OIS Israel and share some exciting technologies coming out of this great country. Thank you very much for your time.